record. Okay. Now let's go back to, okay, now I'm recording. Okay, so you've got the dendrites, which are the shorter branches from your neuron body. And sometimes the word soma is used for the body, but you might just see body or soma. Either will refer to a body as something, the central portion of something. So the dendrites are afferent. Okay, they are incoming. They receive incoming information. Usually they're short and branched. Sometimes the dendrites look like an axon, and we'll talk about that in a minute. Then you've got the body of the the neuron with a very large central nucleus. You probably recognize this picture over here, this purple one, when we looked at tissues. Gosh, within the first 3 weeks, I think, or something like that. So dendrites are not myelinated like you think with the axon, but the axon is myelinated, most usually. Just I never say never and never say always, but usually most neurons or axons are myelinated. So you've got these little Schwann cells that cover certain areas. So those are referred to as the internodes. The bare spots between are the nodes of Ranvier. And if you remember, you've probably talked about the action potential of muscle, but not quite nervous yet. But your action potential actually jumps from node to node. These insulated areas where the Schwann cells are prevent that from happening, under the Schwann cells. So the action potential actually jumps from node to node. So it makes it much faster. So a myelinated axon conducts that signal a lot faster than an unmyelinated axon. At the axon terminus, you will have the synaptic bulbs. Excuse me. So just again, know the dendrites usually short and multi-branched, axon usually singular and very long. Again, in lecture you'll probably talk about it more, but the axon hillock is the area where the axon kind of spreads out and joins the body, the soma of the neuron, and that's kind of an important area to decide in deciding whether or not a signal is going to continue on down to the axon. Sometimes you get a signal by the dendrites and it's not enough to continue that signal on through the axon. So some of your basic shapes. Now you'll see other areas, other textbooks that might have different shapes of axons that I can't remember it off the top of my head, but I know I've got a textbook somewhere with a fourth axon shape. But you've got multipolar axons, and looks like your textbook has got these same ones. Multipolar, and again, just two distinct regions. It's got the very short, relatively short branch dendrites and a singular axon. Now, sometimes the axon will split off, as you saw, up here where you got collaterals, but in general, there are less axons than dendrites. Bipolar neurons are unique, and you see those in the retina of the eye and in certain areas of the central nervous system. But they're kind of axons that regulate and we'll just say they regulate communication between one region and another. But you usually have this dendrites that are they coalesce into one singular structure as it enters the axon, or it enters the neuron body, and then a singular axon that leaves. But again, bipolars, they have these dendrites that look like axons in a way, but just be aware that dendrites by definition are afferent. They carry the signal towards the cell body. So just don't think that they always look, short and branched, but generally they do. This is the fourth one, the anaxonic. That's right. I knew I've seen some somewhere. So this PowerPoint I'm pulling from is out of Saladin, which is your lecture textbook. Most textbooks I've seen don't consider the anaxonic one, but if you just think anaxonic without an axon, and again, you see these in certain areas in the central nervous system. But I would just be concerned with knowing these top three that are in your lab manual. The unipolar, you've got dendrites again, kind of like the bipolar, they coalesce into one long, and they call it an axon here, but again, it carries that signal towards the cell body. The cell body makes the decision whether that signal will continue on, and it continues off on the other side. Now, structurally, you couldn't tell the difference between the dendrites and the axons in a unipolar. And again, like your lab manual refers to, some textbooks refer to that as a pseudo unipolar. But that you've got this long axon looking structure, and the cell body is kind of off to the side. And you see that a lot of your sensory neurons are like that. So when you guys start talking about the I think it's next week, we'll start talking about the structure of the spinal cord, the dorsal root ganglion at the spinal cord. That ganglion is full of these cell bodies. Okay, so this is typical of a sensory neuron. This bipolar is typical of a neuron that you'll see within the retina or various parts of the central nervous system. And these multipolar neurons are very typical of what you see from motor neurons. Now, your lab manual does not go over a whole lot of the glial cells, but we'll just touch on them a little bit. It mentions neuroglia or glial cells, but neuroglia, they're kind of supporting cells. And I would just, if I asked you anything, I would know no more than what this says here. You've got the oligodendrocytes that form myelin in the brain and the spinal cord, and their cohorts in the peripheral nervous system are the Schwann cells. Okay, so if you're talking about what cells form the myelinated sheaths, if you're talking about a peripheral nerve, it's the Schwann cell. If you're talking about in the brain or the spinal cord, it's the oligodendrocytes. The ependymal cells create the cerebral spinal fluid. Microglia are little, basically macrophages. They are immune cells that eat and destroy foreign matter, dead cells, things like that. Astrocytes, these guys are important because they form the blood-brain barrier. They kind of reach out and surround axons and hold things into place, and form a very important barrier, referred to as a blood-brain barrier, to keep certain things from the nervous system and that travel through the blood. Satellite cells, basically they insulate and help support cells of those certain cells of the peripheral nervous system. So kind of, again, your diagram, again, right out of your lab manual, the, excuse me, the oligodendrocytes reach out and surround the axons within the central nervous system. So this is all central nervous system that you're seeing here. Microglia, little guys, again, they phagocytize bacteria, things like that. Astrocytes, Astra refers to star shape, so they've got a lot of appendages that reach out, and again, they surround the capillaries and prevent things from leaving the capillary that should not be in the brain. And again, the cerebrospinal fluid is created by these ependymal cells. These are in certain areas, like the choroid plexus, that we'll talk about in a little bit. And they, kind of like some of the cells within the kidney, they filter blood and let some of the fluid portion of blood out to create cerebrospinal fluid. So cerebrospinal fluid, just like urine, is a blood filtrate product. So again, in the peripheral nervous system, here's how your Schwann cells kind of work. They're basically a single cell with one nucleus, and the cell membrane kind of wraps several times around the axon to form a basically an insulation type barrier. So when you talk about the action potential, it jumps from one bare area between Schwann cells to another. So you've got one Schwann cell that wraps around the axon in the peripheral nervous system. In the central nervous system, it's different. The oligodendrocytes kind of reach out, and they can surround several different axons with myelin, but their nucleus stays distinct. Okay, when you're talking about the Schwann cells, their nucleus is there, wrapping around that length of axon. So those are myelinated axons. Okay, there are some axons that are unmyelinated. Now they're not really not totally unmyelinated, but they're myelinated in a different form. So instead of being surrounded by the Schwann cell like you see here, and wrapped around with several layers of the Schwann cell's plasma membrane. They are kind of embedded within the Schwann cell. So there is a little bit of myelin, I guess, around them, but they're technically not myelinated because the speed of conduction is a lot slower in these unmyelinated axons than there is in myelinated. And again, these are these are seen in certain areas, but just kind of be aware there's a difference. Um, let's not cover this slide. This is more for lecture. I should have probably cut this one out. So again, the synapse will cover basic just anatomy here at the synapse. You guys talk about the physiology of what happens here in lecture, but at the end of an axon, okay, the axon terminus or the axon bulb is kind of swollen a little bit, and you have the little vesicles of neurotransmitter. So when that action potential is received down here towards the terminus of the axon, it causes these vesicles to endocytose their contents across this synapse. Now, the synapse is a physical space, sometimes referred to as the synaptic cleft. That's the distance between the presynaptic axon and the postsynaptic dendrite. So the signal travels in one direction. Okay, it travels from the presynaptic across the synapse to the postsynaptic. It never goes backwards. It's always just in one direction. So your neurotransmitter is released, and this is probably representing acetylcholine. It's it's the simplest one basically. Acetylcholine will bind to these receptors, and then those receptors open and allow the influx of sodium. All right, if you remember from your the muscle chapter with the muscle action potential, remember it's the influx of sodium that causes the depolarization. With enough neurotransmitter and enough receptors open, that sodium influx will be enough to trigger the action potential to move on down towards the cell body from that dendrite. So that's basic structure of the synapse. And again, I don't think your lab manual really covers that. It talks a little bit about the action potential, but just know the basics of the synapse. Know that there's a presynaptic that releases the neurotransmitter, and it's the postsynaptic neuron at the dendrite that has the receptors for that neurotransmitter. This is something I do want you guys to know with neuron structure, and this is not really covered in the lab manual, but I think it's important to understand kind of, it's not just one neuron speaking to another neuron. There are various methods of neuron communication, I guess. And again, I like how Saladin diagrams these. So the circle is the cell body, and you've got the axon leaving, and you've got the little triangle, which is the presynaptic the axon terminus, the presynaptic neuron. So you have a diverging system where the axons split, and one neuron can communicate with several different neurons and send that signal to several neurons, who then send that signal on to more neurons. So you have a multiplicative type effect. So you've got the input from one neuron can cause several hundred neurons to fire in response to that as well. So you can multiply your output. You also have basically the opposite arrangement where you have a converging you know type of communication, where you have a lot of neurons that may get stimulated from one area that basically you'll have multiple neurons stimulating like a single neuron, and then these neurons then stimulating a single neuron. So you have a kind of a convergence, just like the name, a convergence of the signal. You see both of those in different areas. But just be aware that there are different types of communication methods. This reverberating one you see in certain areas, and this is where kind of a downstream neuron will have an axon that splits off and then stimulates an upstream neuron, and that kind of keeps that signal going. And that is types of things you see. Not exactly what's involved in like labor and delivery, but I see something like that, and that to me it's almost like a um, Trying to think of the mechanism. Um, it's just eluding me right now, but positive feedback. Yeah, it's like a positive feedback mechanism. So this helps to keep that thing going. So you see that sometimes in certain areas, but like kind of like a positive feedback, it happens until a culminating event, stops that event. And then this parallel thing is just kind of like you'd think with parallel batteries. You have your input is split off, and you can either stimulate a single neuron, multiple neurons, or even more neurons. But eventually, all those neurons coalesce to a single neuron for output. So just if I gave you a diagram of these four types, and again, I would focus on diverging and converging. Those are by far the most common types of setups that you see, but you should just kind of be aware of those. Um, okay, so let's go to see if I can close this and, oh, okay, new share. Let's see that one. I want to do the brain and cranial nerves. Okay. Oh, okay. Make sure I've still got everybody here. Okay. Um, okay, so let's go over the brain and cranial nerves. And again, just to kind of confirm, somebody give me a thumbs up if you see. Oh, no, I don't want the cadaveric photos. What I want this one. Sorry. Okay. Why am I Dutch? Okay, now, all right. So does somebody see a big picture of a brain with what looks like a nasty looking tumor? Okay, good deal. Yeah, I don't know exactly what type of tumor this is. I just downloaded the photos, but yeah, this is a mid-sagittal section of a brain with a pretty nasty looking tumor in the occipital lobe. Um, so just, yeah. Now, like the muscles, I will give you guys a list of terms. Again, I've been working my butt off just to get to this point. But by the end of the day today, I will put on Canvas a list of terms that you want to know. But I will tell you right now, a lot of them are going to be on this first one, with as far as structures of the brain and things like that. So with the lobes, you want to know the various lobes, and again, there's not a real hard and fast boundary. I'm trying to see. I thought I had one that was a color photo of the lobes. Maybe not. Okay. I'll try to get I've got one somewhere that's like a color photo of the lobes, but for generally the lobes are going to be deep to the bones with the same name. So you've got the frontal lobe and the cerebrum's kind of up front, the parietal lobe kind of in the middle, the occipital lobe kind of in the back, and the temporal lobe would be kind of here under the temporal bone. Now you do have a lobe that is referred to as the insula, or the insular lobe. Uh, and trying to think, I can't remember. I'm not seeing it on your lab manual, but the insular lobe is deep to this temporal lobe. You actually have to kind of peel this back and look deep within this lateral sulcus to see that insular lobe. But I've got another PDF here. It's got it diagrammed as well. The other thing you want to know is the pre and post central gyrus. Now, you'll probably talk about that in lecture, but those are important areas because those are where the motor and sensory cortexes are located, which basically, if all the sensory in your certain areas of the body is picked up on one of these lobes, and all the motor is picked up on the other, it's kind of interesting. There's a little structure called the homunculus or little man that they use to diagram those. Again, that'll be more of a lecture thing if they cover that, but we're not going to talk about that. So your cerebrum is the large portion that you see here. Um, so you've got, again, your left and right cerebral hemispheres. They are split down the middle by the longitudinal fissure. Okay, there is a uh, lateral sulcus that splits the temporal lobe from the frontal and part of the parietal lobe, and you have a transverse fissure that splits the cerebrum from the cerebellum. Okay, so the cerebellum is just kind of deep and underneath the occipital lobe, and you have along kind of the brainstem, again, the pons and the medulla. And these are basically just swellings that sometimes it's really on a brain it's hard to distinguish the two, but usually if you're looking at a diagram you'll see a cleft between the two. So the pons will be the more superior and the medulla will be the more inferior of the swellings, and going in down into the leading into the spinal cord. um, yeah, so looking at the surface of the brain, there'll be others showing this, but the gyri are the surface folds, I guess. If you think about gyrus and sulcus, the sulcus are the valleys that you see, and the gyri are the mountain tops. If you kind of think about that with what you see, so when you're looking at the brain, the postcentral gyrus is the surface area that you see just behind the central sulcus. And the sulcus, again, the sulci are the valleys, and the gyri are the mountain tops between the valleys. If you think about the surface area of the brain. You should be able to recognize the mid-sagittal section. By and large, the quiz, practical, whatever it is, however it is, I end up questioning you guys on this. I will definitely have a mid-sagittal section. So I think important things to know over here are the corpus callosum, which is this large structure here, kind of in the middle. Um, I guess I haven't asked this, but I'm just assuming you guys can see my mouse as I'm moving this, right? Can you guys see my mouse? No? Okay, thanks, Mallory. Appreciate it. Okay, I just thought to ask that. I've been trying to do narrated PowerPoints with the little laser pointer thing they have, and for some reason they don't record. So the laser pointer doesn't record on a Mac, but I guess it does on Windows. So I just thought I'd ask. Okay, so the corpus callosum here, this large structure, this is a big bunch of fibers that connects the left and right hemispheres of the brain. Okay, so this is your communication between the left and right halves. Under that, you have the thalamus, and just deep to that, you have a region called the hypothalamus. And then next to the hypothalamus, you have a few other structures. The optic chiasm, that is where the optic nerves cross halves. Part of that optic signal crosses to the other half of the brain. And then just coming off of that, you have I don't see it labeled the infundibulum here, but you have the infundibulum is the stalk that leads down to the pituitary gland. Okay, so some of these midline structures you only see when you've got a mid-sagittal cut. I'll go ahead and talk about it here since it's here, but I've got other diagrams showing it later. The other things you want to know are, if you look on, if you look on page 166 actually it's 169 on exercise 14 in your lab manual, they show the ventricles. So the brain has got these fluid-filled areas inside referred to as the ventricles. The lateral ventricles, ventricles 1 and 2, are more located in the cerebral hemispheres, but you can see a little bit of where they connect, they really connect around the thalamus. So you don't really see the ventricles labeled on a mid-sagittal section, at least not the laterals, but on the mid-sagittal section, you will see usually at least the fourth ventricle labeled. Now, the fourth ventricle sits between the cerebellum and the pons, and that leads down into the spinal cord. Now, the spinal cord has got a central canal as well, so this is all for the flow of cerebral spinal fluid. So cerebral spinal fluid flows through the deep parts, the inside of the brain, as well as surrounding the brain outside to kind of cushion it. So I don't see a label here for the third ventricle, but if you look on page 169 in your lab manual, you can see the third ventricle, and again, it kind of surrounds this area of the thalamus. But the cerebral aqueduct, sometimes you'll see referred to as the aqueduct of Sylvius. Um, but your lab manual just refers to it as a cerebral aqueduct that carries cerebral spinal fluid from the third ventricle down to the fourth ventricle, this nice opening between the cerebellum and the pons, and it continues on down into the spinal cord. And you do have an aperture here, which if we looked at the bottom diagram on page 169 in your lab manual, you have the median aperture, and that allows cerebral spinal fluid to go from the deep parts of the brain to the area surrounding the brain. So when you think about somebody with hydrocephaly, sometimes this aperture is closed or doesn't allow enough cerebral spinal fluid through. And cerebral spinal fluid is produced within the deep parts of the brain in the choroid plexus, which you actually see a little bit down here in this mid-sagittal split of a real brain. So if you look like where my cursor is, there's a dark area here and a dark area there. So that kind of leads into the lateral ventricles. Again, the lateral ventricles are off the midline, and what we see here is a mid-sagittal cut, which is right on the midline. So you don't really see non-midline structures in a mid-sagittal section, but you can kind of see that they do go off of that midline. So the choroid plexus is where that fluid is created, and it's reabsorbed. I'm blanking on where it's reabsorbed, but anyway, just be aware that it's, choroid plexus is full of those ependymal cells that we talked about earlier, and that's where you see the CSF being created. So let's see some other things in the midline. The pineal gland is very important. It's part of what you referred to as the epithalamus, which is kind of a new structure. When I was sitting where you guys were, 20 years ago, it was just the thalamus, the hypothalamus, midbrain, pons, medulla, etc. But the epithalamus is kind of a newer thing. That the pineal gland is what they're calling at least the big part of the epithalamus. Don't worry about the habenula, but the pineal gland, that's important because that creates serotonin, and that regulates our sleep cycle. That's our third eye, as sometimes you'll hear it. Now, it's not labeled right here, but the surrounding area right there I'm looking at, that is part of the midbrain as well. That's your inferior and superior colliculi. I'm sure you'll probably talk about that in the lecture, that some reflexes are centered around there, like when a loud noise, it makes you turn your head and look towards that direction. That is a reflex that correlates sound and sight as well. So those are some important things you need to know about the midline structure of the brain. With something like this, you want to know the layers of the dura mater, or the meninges. The dura mater is the outer layer, the arachnoid mater is the middle layer, and the pia mater is the innermost, deepest layer. So I may ask you a question or two about that, but I would just, don't worry about labeling this diagram, but I would know that the dura mater is the tougher outer layer. I won't ask you about the two, layers of the dura. Just know the dura versus the arachnoid versus the pia. And the arachnoid mater covers the brain stems, or the vessels of the brain, but the pia mater does not. It actually, the pia mater goes down into the sulci as well. So the pia mater is very intimately associated with the brain. It dips down into each of these sulci, but the arachnoid mater has this kind of like cobweb like webbing that creates a space between the membrane and the pia mater. And it's actually, now that I see it, I remember now, it's the arachnoid granulations that reabsorb the cerebral spinal fluid. That's where it actually is reabsorbed. But just know the three maters. Okay, I thought I had one. So here is a diagram. You see this blue here on the brain. This is kind of a diagram of the space that is your ventricles. So here you've got the two lateral ventricles. Again, they extend out pretty far off the midline, and they kind of look like ram's horns in a way. If you think about the shape, there's a little bit that goes interior, but most of it goes kind of curves posterior and goes out laterally. And these actually extend out into the temporal lobes. Then you've got like the midline structure. So the there is no first and second ventricle, if you notice. You just have the left and right lateral ventricles. Those are ventricles 1 and 2. The third ventricle is on the midline, and you've got the cerebral aqueduct connecting the third ventricle to the fourth ventricle. Okay, and doesn't miss me. All right, and then it continues on again into the central canal, the spinal cord. So a lot of these diagrams, you'll see a brain slice in a certain plane. You should see a brain next to it with a little glass plane. This is showing that the section or the region where this section is taken. And if you see, you've got the lateral ventricles. This is the one that kind of extends anteriorly out this way, and then come down here, and it would extend back out and around into the temporal lobe on each side. And within those lateral ventricles, you see these little, they almost look like, I don't know how to describe them, but it looks like little fuzzy material. But this is a very vascular structure. Again, that's the choroid plexus, where the blood vessels are surrounded by ependymal cells, and they filter the blood and turn the liquid portion into the blood into cerebrospinal fluid. And you see your longitudinal fissure. You've got the various lobes, things like that. In this diagram, pretty much almost verbatim what you see on the bottom of page 169, showing you kind of the flow of cerebrospinal fluid. Again, it's created within the choroid plexus, deep within the brain, flows through the ventricles, and then out and through the median aperture into the subarachnoid space. And in those arachnoid granulations kind of that stick out between the arachnoid motor that you saw, and right here, the arachnoid granulations almost look like, they remind me of little mushrooms when they stick out through the dura mater and back into the kind of the bloodstreams where it the CSF is reabsorbed in the venous blood. So, and usually when you see hydrocephaly, usually that's I don't know exactly what causes it. I'm sure there's various things, but I think it's usually like a restriction of the aperture. Then the fluid buildup within the brain makes it enlarged, and you usually see that in babies because their skull is still soft. I mean, if that occurred in adults, it'd be traumatic because you've got a pretty rigid cranium at that point. I don't know that I'd really ask you much on these photos, but I do think they help in a little bit. One, in visualizing where the cranial nerves actually exit the different areas of the brain. So basically what you have here is you've got the thalamus. I don't think you actually have the hypothalamus kind of on this. The hypothalamus would be, I don't really think you see the hypothalamus on this structure. So really you've got like the midbrain, the thalamus is blue, midbrain is red, pons is kind of a lighter green, and the medulla is a darker green. So basically if you could, go to like a mid-sagittal structure like this and basically just pluck off the cerebrum, you would see something like this. But I do think this helps visualize where all the cranial nerves kind of come off of the brainstem. And most of the cranial nerves are straight from the brainstem, with the exception of the optic nerve and the olfactory nerve, which is number one. You don't see that because that is actually under here, under the frontal lobe of the cerebrum. So I wouldn't particularly ask questions on this, but I do think this structure helps you visualize that as well as where the pineal gland is located in relation to the midbrain. Remember I talked about the superior and inferior colliculi, these pair of bumps on the posterior portion of the midbrain. So let's see what I want to get out of this one. Basically with this, I want you to understand that the thalamus and hypothalamus are what they sometimes refer to as relay stations. So you've got this sensory information coming up the spinal cord, and it goes to the thalamus, and there's a decision made on like where to send that information. Is that what part of the brain does that go to? So there's radiating fibers that kind of leave out from the thalamus to other parts of the cortex. And then thalamus makes, there's a decision made, and it comes back through the thalamus, and it leaves through the motor fibers. So I just wanted you to get a generalized picture of things like that, so that you see in the lecture and stuff, the thalamus is almost always referred to as like a relay station of some sort. Again, just kind of a closer look of the cerebellum and what they refer to as the arbor vitae. Arbor vitae is actually a kind of tree. It is it's an evergreen shrub bush type thing that gets really big. But why this is called the arbor vitae in the cerebellum refers to the white matter. So if you look at the white matter in the cerebellum, it kind of centralized here, and then leaves, and it branches out in these really thick branches. Now, you guys should also know, hopefully you guys have hit it in a lecture if you haven't yet, white versus gray matter. White matter is anything that's myelinated. So when you look at it under a microscope, it's white as opposed to grayish. So white matter is myelinated axons, and gray matter is anything that is unmyelinated. So gray matter is either cell bodies or unmyelinated axons. Okay, so just general trends. If you see white matter, that's myelinated axon. So white matter is going to be communicating type in a way. So you think the myelinated axons, that's the wire that carries that signal away from the cell body or towards the cell body. And the gray stuff is stuff, the gray matter is stuff that's going to be making the decisions. That's cell bodies and dendrites. So again, white matter is myelinated axons, gray matter is dendrites, cell bodies, and unmyelinated axons. And again, you've got the kind of relationship here between um, the ocular motor nerve, the pons, medulla, and your cerebellum, as well as your fourth aqueduct. And then here it's not labeled, but you can see the median aperture just inferior to the cerebellum. Don't worry about the structure here, the cerebellum, this superior view. But you do have this weird little structure on the midline called the vermis, which is the worm. It's just kind of a long lump type thing on either side. If I were going to ask you something about the cerebellum, it would probably be the arbor vitae. Again, I don't think I'll ask you much on this, but you may hit more on this in lecture. There are various types of fibers. Now, in this position, or this diagram here, the tracks are basically bundles of axons. So you think about communication within the brain, you've got these association tracks that connect one part of the brain to another part of the brain. The corpus callosum is, again, right here in the middle is what they refer to as commissural tracts, and that connects the right half and the left half. And the green that you see up here at the top and down here are what they call projection tracts, and they basically project up into the brain and connect certain areas of the brain with the spinal cord. And if you notice, there's an area here just deep to the and it's in the middle of the medulla, but it's just superficial, or sorry, inferior to the pons, is where they have what's called the decussation in the pyramids, and that's where the projection fibers cross from one half of the body to the other. So if you always, I'm sure you've heard before, the left half of your brain controls the right half of your body, and vice versa. That's because of this decussation here in the pyramids. It's where the fibers cross from one side of the body to the other. Again, this photo here, I won't ask you about the nuclei and things like that, but I just thought this was a good photo to again and visualize the lateral ventricles and how they surround the thalamus. And yeah, the little structures here will be your oh gosh, I'm just blanking. Where the cerebrospinal fluid is made. But anyway, so yeah, you've got your thalamus here on either side, and in your kind of third ventricle between, again, pituitary gland is a midline structure, and here's your optic tract. So what you're seeing here, and again, if you look, you got the plane of this section. If this section were a little bit more posterior, you'd see where the optic tracts would actually be a single structure on the midline, because when they cross, they kind of form an X. So this is, you know, kind of anterior to where they'd cross. But if this section, again, you go over here, if you go a little bit farther back, you would see these two structures here would come together, and they'd form the optic chiasm, where there's, you know, where they cross on the midline. And yet here, yeah, you also see the insular lobe here. So if you look at this this sulcus here, this lateral sulcus, if you open that up, you can see the insular lobe um, that is deep to your temporal lobe. Again, you don't really see that insular lobe on a surface photo of the brain. Okay, so with the cranial nerves, I may give you a diagram like this, and have you label the cranial nerves. Now, if I do that, I will probably have maybe a couple of them labeled so you have some reference of some sort. Right now, I will not ask you to associate the name and the number. Okay, when I was in school, it was as big in knowing, you know, the name and what number it was. I'm more interested in you knowing the name and where it leaves the brainstem, and maybe knowing a little bit of something about it. I think the acronym I learned was on old Olympus, towering tops, a Finn and German viewed some hops, or something like that, which doesn't really work now. Oh, Finn and okay, so when I knew this, a Finn and yeah, okay, so and was this used to be the vestibulocochlear nerve, used to be called the auditory nerve. That's why I knew that. It's and German viewed, and then it goes from 10 to 11 some. This accessory nerve used to be called the spinal accessory nerve, and then hops. Well, yeah, so that my acronym doesn't even work anymore because the names have changed. But I remember the stupid acronym for some reason. I don't know. But if that type of stuff helps you, you know, whatever. But I feel like if you know the name, it'll tell you something about what each of these nerves do, or something about them. The olfactory bulb, that's your sense of smell. Okay, if you notice, it goes up to the underneath the frontal lobe. This is hanging in the roof of your nasal cavity, and that's where you pick up smell. Optic nerve is the sense of sight that comes from the eyes, crosses, some of those fibers cross the body and go to the opposite side at the optic chiasm. Oculomotor nerve, it tells you oculo, eye, motor, controls the movement of the eye. So this controls some of the movement of the eye. There are actually three muscles that control eye movement: oculomotor, trochlear, and abducens. And if you think about the name oculomotor, it tells you it's motor control of the eye. Trochlear nerve, this goes to a specific muscle of the eye called the superior oblique that hooks through a structure called the trochlea. Hopefully you guys remember the trochlea as a distal part of the humerus. Remember the trochlea and the capitulum. The trochlea articulated with the ulna, and the capitulum articulated with the radius. But the trochlea, and this is like a hook type structure, or a loop that changes the direction of that muscle. I'll show you in another diagram here in a bit. That's why I remember the trochlear nerve. Trigeminal nerve, it's got three large branches that goes out to a lot of the parts of the face, and we'll talk about the different branches here in a minute. Abducens, again, that goes to an eye controlling muscle that abducts the muscle. Or abducts the eye. It pulls the eye away from the midline. Facial nerve goes out to the face. There's a lot of sensory carried on the facial nerve. vestibular-cochlear, the vestibule, and the cochlea. The vestibule picks up balance information from the vestibule of the ear, which is kind of the opening leading into the cochlea, and the cochlea itself picks up sound. So the vestibular-cochlear that I used to know as the auditory, vestibular-cochlear is a little more descriptive because it's not just sound, it's balance as well. So the semicircular canals as well as well as the cochlea. So you pick up balance and auditory information from that. Glossopharyngeal, glossus refers to the tongue, pharyngeal refers to the pharynx. So this is movement of the tongue and your pharyngeal constrictor muscles that allow you to swallow and move your tongue and controls a certain part of the tongue as well as the hypoglossal nerve. Vagus. The vagus nerve controls a lot. This goes down into the thorax and abdomen. And some of these nerves too, I probably won't have you specifically know whether they're pure motor, pure sensory, or a mix. But some of these nerves are pure sensory. Like, it should make sense. The olfactory bulb is pure sensory. There's no motor that, control that goes out. You don't have anything motor in the eye. The optic nerve is pure sensory as well. The ocular motor is obviously pure motor. It controls things in response to other things. But the vagus is one that's mixed, definitely. It picks up sensory information as well as motor control. And the big thing I remember about the vagus is that it controls heart rate. And I know some of you guys are going into nursing. I'm not sure if I told you or not, but I was in a nursing program long ago when I first got out of school, not really knowing what I wanted to do. But I spent my time in a nursing home and as a student nurse. And one of the things I saw was something that never should have happened. When you have older folks, sometimes they have trouble going to the bathroom, and they gave enemas and things like that, but you're never supposed to do what they refer to as a digital rectal stimulation. Basically, you stick a finger up the anus and you trigger the vagus nerve. The goal there is to stimulate a bowel movement, but the other thing you can stimulate with the vagus nerve by sticking a finger in the anus is you can cause the person to quote unquote vag down, which is the vagus nerve stimulates the heart rate to decrease. So if you can stimulate the heart rate through the anus, and with these older folks who have blood pressure problems already, that can cause a dangerous lowering of the blood pressure. So you never actually should do that. Just FYI, something that you didn't know you can control the heart rate through the anus, but you can. Hypoglossal, again, below the tongue. So this is some of the deep tongue muscles and pharynx muscles. And the accessory nerve, I used to know that as a spinal accessory nerve. Now that is one that you see here. It actually goes down the brainstem and comes out from the same area. Now, if you see these guys here that aren't labeled, these guys are actually spinal nerves. So the spinal accessory originates from true spinal cord outside the foramen magnum. It goes up into the foramen magnum and then loops back out through the jugular foramen to come down. And the spinal accessory acts more like a spinal nerve. It controls the sternocleidomastoid muscle and trapezius muscle, which is kind of odd, but that's what it does. Okay, no worries. If you have to leave, take off, do what you need to. This is recorded and will be put up on Canvas. So FYI, I'm not going to go through these. I've already hit on all these, but basically go through and be able to. I wouldn't ask you to give me the function. What I would do really is give you the function and ask you the name of the nerve. Okay, so I'm going to do it what I think is the easier way. I'll give you the information and you tell me what nerve I'm talking about. But you guys should be able to go through all the olfactory, the optic, all these cranial nerves and tell me what nerve does what function. So again, ocular motor, you're looking at only specific muscles. I think you're looking at 3 of the 6 or 4 of the 6. Superior medial and inferior rectus, inferior oblique. So 4 of the 6. And the other one is the trochlea. you've got this the superior oblique muscle, which goes through a little trochlea, a little loop-like structure to allow it to change direction. Yeah, here's the trochlea, this little thing right here. All right, so you got this muscle that goes up and loops and changes direction. So there's the superior oblique that goes through that. So that's why I remember the trochlear nerve is associated with the trochlea. The only other trochlea I know of is at the distal end of the humerus. Trigeminal, I would not ask you about the different divisions, but just know that it's mostly sensory. There's some mixed. You look at some motor, but just again, just be able to identify these nerves if I gave you some sort of identifying information about them. Okay. Again, don't worry so much about the different branches. Again, the facial has a lot of different branches as well. There's 5 different branches of the facial. Again, it tells you kind of where they terminated: the temporal area, zygomatic area, the cheek, the mandible, or in the neck. And again, vestibular cochlea. The cochlea is the snail-like organ that picks up hearing. The vestibule is the opening to it that with the semicircular canals picks up balance and motion type of information. And then again, glossopharyngeal is the tongue, some of the pharyngeal restrictors. Vagus, you should know the vagus. That is one that goes, again, out to just about everything in the thorax and abdomen. It's a very important one. And again, the spinal accessory is kind of a unique one. It's odd in the way that it originates outside of the cerebrum, side of the cranium, but it goes up back into the cranium through the foramen magnum and loops back out through the jugular foramen to control skeletal muscle. Okay, so that's kind of unique amongst cranial nerves. You think most of the cranial nerves don't really control, well, skeletal muscle in the face, but skeletal muscle in the neck and shoulders is that's unique amongst the cranial nerves. Again, here's kind of a generic diagram of telling you whether sensory, motor, or mixed. But again, I will ask you kind of identifying information about the cranial nerves, and you just tell me what they are. So the other thing I'm going to put up on PowerPoint that I will probably use as a as like testing information. I'll probably use diagrams out of this for the practical, because I like these because they're color coded. You guys see a picture of the brain with big orange photos, right? Somebody give me a thumbs up. Let's see. Yeah. Okay. So Mallory, you do see the big orange brain. Okay. All right. Good deal. Like I said, I just want to make sure you guys see what I see. So I've got this PowerPoint here of the brain, and I like this because they, not just like the cartoons, they have the area actually highlighted in a certain color, and it's definitely so you can definitely tell like midbrain versus pons versus the medulla. I like these, and they got pictures of these are actual cadaveric brains, and they have the nerves highlighted. So I would go through this one as well. Again, I won't ask about the certain specific parts of the cerebellum and things like that, but I just wanted you to see that the cerebral aqueduct, you do have this opening between the cerebellum and the pons, which you kind of see here. So I will put these up on Canvas. I think I got one of these uploaded already before we started our little talk, but I will put these guys up on Canvas. And just kind of a reminder, remember you've got there's another quiz on the muscles on the lower body that's due. I will put these things up on Canvas. And I think what I also want to do is I want to make an assignment. I want you guys to finish, and I'll try to scan these and put them up on in case somebody does not have their lab manual. But I want you guys to do the review sheet for exercise 13 and 14. So that's basically going to be pages 159, 160, 161, and 162. And then for exercise 14, it'll be 175, 176, 177, 78, and we're not going to do 79. That's dissection of a sheep brain. We don't have that, but I want you guys to do that as well. And I will make that a quiz grade or something. I'll make it worth it. But I want you guys to do that on a text document or Word document, something, and just, you basically kind of recreate what you see there. You can just give me, question number 1, 2, 3. I think it just goes 1 through 16 on chapter or exercise 13, and then exercise 14. It's just 1 through 9. Yeah. So I will put that up on Canvas as actual an official, assignment. But I just thought I'd have you guys do that, and I'll make that due by midnight next Thursday as well. So I'll give you guys a week to do that. And I may put another quiz or 2 up as well. But like I said, I'll give you guys plenty of things to kind of keep you busy, helping you guys with this as well as lecture. But I think that this will also give you guys more opportunity to bring up grades and things like that. So I feel like things are challenging enough as it is. I don't want anybody to get a poor grade because we went to this kind of mode of learning. But just kind of keep that in mind. I want you guys to do the exercise at the end of exercise, or the review sheets at the end of exercise 13 and 14. And again, don't forget about the muscle quiz that's due, I think by midnight Saturday, if I'm not mistaken, the 4th. Okay.